Hello to everybody. In this presentation, I will talk about the sources of variation in vegetative and reproductive traits in Plantago lanceolata. Databases of functional traits, such as try or Fred, are commonly used to infer trait-environment relationships, but very frequently these trait datasets are observational. That is, traits were recorded from in situ field populations. And in this sense, it is very important to evaluate its potential uses and misuses. For example, if we find correlations between trait and environmental data, can we infer genetic differentiation from trait-environment relationships? This is important because observational data may result from both genetic differentiation and phenotypic plasticity, and plasticity might obscure trait patterns driven by genetic differentiation. Common garden experiments are very useful precisely for disentangling these genetic and plastic responses. And by growing offspring from multiple sources populations together in a set of controlled treatments or exposure environments, we can tease apart genetic differentiation and plasticity. In these plots, we can see model examples and model examples on how source and exposure environment determine trait values across populations. In the horizontal axis, we have the environmental conditions of source populations, for example, from low to high precipitation, and each of the blue lines correspond to one exposure treatment. For example, here, low and high water availability. So here we see, for example, how populations, we can see the trait values from populations with a low source precipitation to populations with high source precipitation. But in this case, those when they are exposed to the low water availability. And here the same, but when they are exposed to high water availability. We can also plot the expected pattern to be observed in the field with these dashed lines. And interestingly, depending on the magnitude, direction, and interactions between source and exposure effects, there will be more or less correspondence between genetic differentiation and what we actually find with observational data. With stronger source effects, with a coincidence in the direction, for example, here with a stronger source than exposure effects, with a coincidence in the direction of source and exposure effects. For example, here, trait values increases from low to high source precipitation, and also increases from low to high water availability, exposure water availability. So with the same direction in the source and exposure effects, or with source effects whose direction is independent from the exposure environment, we will be able to infer genetic differentiation from observational patterns. For example, populations from water environments would be genetically determined for higher values in the studied trait. And we would actually find this pattern using observational data. In contrast, with stronger exposure, with a stronger exposure than source effects here, exposure than exposure than source effect, sorry, or with opposite direction of source and exposure effects. Here we have an increase in trait values from low to high source precipitation, but we have a decrease in trait values from low to high exposure water availability. Or with an interaction between source and exposure effects that leads to a direction in source effects that depends on the exposure environment. With those characteristics, we won't be able to infer genetic differentiation from observational patterns, because plasticity in this case will be modifying or masking the patterns. And following the example, we could have the case of no genetic differentiation, but we could find a positive response if we use observational data, just because the existence of plasticity. So, okay, to summarize the rationale of this study, the idea is that first, by comparing the magnitude, direction, and interactions of source and exposure effects, we can disentangle genetic and plastic responses. And second, by comparing the trait patterns detected in the greenhouse with those detected with field data, we can evaluate the potential uses and misuses of observational data sets, and in particular, their usefulness for inferring genetic differentiation. So to achieve these objectives, we monitor multiple populations from Plantago lanceolata, spanning a large environmental spectrum and including both native and non-native ranges of the species. In 46 field populations, we recorded plant biomass, SLA, flowering, yes or no, and in inflorescence length as a proxy for fecundity. And we also obtain climatic and land use data. In a common garden experiment with offspring from a subset of 15 populations of those 46, a subset of 15, we recorded the same traits as well as root-to ratio, and we expose plants to different levels of water and light availability. So now I will try to summarize the main results. And here we can see trait responses to source and exposure environments. And as in the conceptual figure that we saw before, we have a source environment. In the horizontal axis, we have aridity, temperature, vegetation cover, and mowing. And exposure environment, which are the water and light treatments, and which are also depicted with different lines with different colors. So one of the main results is that vegetative and reproductive traits show contrasting patterns regarding genetic and plastic responses to environment in the common garden experiment. Here I show specific leaf area as an example of a vegetative trait, and whether plants flower or not as an example of a reproductive trait. And looking at the three characteristics of source and exposure effects that we mentioned previously, we found that vegetative traits show in general stronger responses to exposure environment. So we can see the stronger separation between lines. We also show, compared with reproductive traits, we also found opposite directions between source and exposure effects. For example, here, in this case, we can see a positive effect of aridity on SLA, with a positive effect of source aridity on SLA, but we find a negative effect of diminishing water availability in the greenhouse. And we didn't find similar examples of this phenomenon in reproductive traits. And also we found in vegetative traits interactions between source and exposure environments, so that we can see here these changes in direction in the source effects. Okay, so this difference, and well, we can see the opposite pattern in reproductive traits. We see smaller, small exposure effects. We see the direction of source effects is the same irrespective of the exposure environment. We see some changes in slope, but no changes in direction. So these differences apply to all vegetative and reproductive traits analyzed in this study, but well, we don't have enough time to go in detail in this presentation. But anyway, the stronger role of genetic differentiation in reproductive traits probably arises because of the more direct relationship with fitness. In contrast, traits less directly related to fitness, like in this case, the vegetative traits, would instead display higher plasticity, and this would be to buffer short-term environmental perturbations and ultimately to maintain fitness homeostasis. And now we can compare experimental and observational field data. In this table, we can see the effect of source environmental drivers on traits detected with observational field data and in the greenhouse, and this one being genetic differentiation. So we have, as I said, yes, the effect of source environmental drivers on traits. And for example, there is a positive effect of vegetation cover on biomass when looking at observational data. And the effect is not clear when looking at genetic patterns because of a presence of interactions between source and exposure effects. That's what it's meant with that sign. And there is an absence of effect of cover on SLA in observational patterns, and a lack of clear effect when looking at the genetic pattern. Okay, in the comparison column, we show a green square when there is consistency in the presence and direction of effects between observational and genetic patterns, and a black square when there is no consistency. And here the main result is that there is consistency between observational and genetic patterns in reproductive traits, but not in vegetative traits, as can be seen from the majority of black squares in the vegetative traits, and as opposed to the presence of always green squares in the reproductive traits. Okay, and these different patterns arise from the previously mentioned differences found between vegetative and reproductive traits in their genetic and plastic responses to environment. So after presenting the results, we can have some important considerations to make. And the first one would be that species life histories may determine which traits are most closely related with fitness and thus show stronger genetic differentiation. In herbaceous plants with short lifespans, demographic theory predicts reproduction to be the most influential rate on population growth. However, longer-lived plants, such as trees, could show stronger genetic differentiation in traits related to survival, since survival is more influential on population growth in species with longer lifespans. Okay, this could be something to test in the future. And the second consideration is that we found in Plantago lanceolata that reproductive investment was highly dependent on biomass. And thus we included biomass as a covariate in all analyses of reproductive traits. Interestingly, when we repeated the analysis without including this covariate of biomass, reproductive traits were no longer showing different patterns than vegetative traits. And this highlights that reproductive traits can be partitioned into two components: biomass, which is very plastic, and reproductive investment per unit biomass, which is instead strongly determined by genetic differentiation. To finish, the main conclusions of the study were that the ecological and evolutionary mechanisms underlying Plantago lanceolata trait variation differ between reproductive and vegetative traits, being reproductive traits determined by genetic differentiation and vegetative traits by plasticity. And these differences seem to arise from the relationship between each type of trait and overall fitness. Second, as a consequence of this, there were some inconsistencies between each type of trait and there were some inconsistencies between observational trait-environment relationships and genetic differentiation in vegetative traits. Third, observational data may reliably inform about the species' evolutionary potential and the drivers of selection only for the traits most closely related to fitness. And fourth, we also advocate for considering biomass dependency in trait variation analysis, as well as the implications of species life histories on trait fitness relationships. And I think that's it. Thank you very much for listening.